Hello everyone. In the last class, if everyone quickly recall, we have finished the hormone abscisic acid and ethylene. So what are the various hormones that we have completed so far? We have completed the hormone auxin, we have completed the hormone gibberellic acid, we have completed the hormone abscisic acid, and we have completed the hormone ethylene also. What are the hormones that we are left? We are left with the hormone cytokinin, jasmonic acid, salicylic acid, brassinosteroids, and little bit we have to also read about the polyamines. So in this particular class, I am going to complete the entire hormone of cytokinin. Okay, all of you listen to me very carefully. Friends, if every one of you quickly recall, whenever we are studying the hormone auxin, I told to all of you to remember that auxin is the rooting hormone. Is it? Of course, yes. Why? Because friends, auxin is actually synthesized in the shoot. From the shoot, it is getting traveled to the root. We all people commonly know about that. Now listen to me correctly. But when you are reading the cytokinin, friends, what we can say that the hormone cytokinin is the shooting hormone. Why do we call cytokinin as the shooting hormone? What will be the reason? The reason is very simple, friends. Cytokinin hormone is getting synthesized in the root. From the root, where it is traveling, friends, the cytokinin is traveling towards the shoot, and it can show their action completely in the shoot. Not to get confused with that point. So now I am going to explain you a little bit about the discovery of the cytokinin, how the cytokinin got discovered. All the students try to understand my statements very correctly without confusion, friends. If the cytokinin hormone want to act in the plant body, it is compulsory, it is mandatory, it is necessary, friends. There should be a presence of the auxin in the plant body. What I'm trying to explain you, if the hormone cytokinin wants to act in the plant body, what is compulsory? Auxin is compulsory. Okay, let me explain you, friends, how the cytokinin got synthesized. What the scientist has done, every one of you listen to me correctly. They have taken some mixtures. They have taken coconut milk mixture, or they have taken vascular extract, nothing but they have taken the xylem extract, not to get confused with that particular point. What they have taken coconut milk or the vascular or the xylem extract, or they have taken the DNA extract. So friends, what they have done to these three mixtures, all of you has to understand me correctly. To these three mixtures, what they have added, friends, they have added the hormone auxin. What they have done, they have taken coconut milk or vascular extract or DNA extract, and to these three mixtures they have added auxin. And what they have done, they have introduced, where they have introduced this entire mixture, they have introduced this entire mixture in the internodal region of stem of the tobacco plant. See friends, this is the stem of the tobacco plant, you know, right? These areas are called as what, friends? These areas are called as the internodes. Exactly, friends, we have introduced all these mixtures along with the auxin in the internodal region. Friends, then you know what happened? It starts showing the division. What starts showing the division? Especially the cytoplasm starts showing the division. So that time they understood, what the scientist understood. So all these mixtures, listen every one of you, listen every one of you, all these mixtures contain a chemical, or any of these mixtures contain one chemical. That chemical, when combined with the auxin, it is promoting the cytokinesis. So friends, what is that particular chemical that is isolated first? All the students listen. Coconut milk was found to contain one particular substance. What is that particular substance is helping? It is helping the plant to remain in the cell division cycle for the long period of time. You know, friends, from where the first cytokinin was identified, all the students has to listen to me. First cytokinin was isolated from the herring sperm DNA. What is herring sperm? Name of the fish. First, from the fish, we have isolated the cytokinin. You know, friends, what is the name we have given? We have given the name Kynutin. Not to get confused with that, friends. The first cytokine was isolated artificially from the herring sperm DNA, and we have given the name kynutin. Not to get confused. Later, all the students listen to me very correctly. The same scientist Miller, the same Miller isolated the cytokinin, listen every one of you, from the immature endosperm of the corn. And he has given the name called as giatin. Most of the students are getting confused, friends. Naturally occurring cytokinin is called as the giatin, which is taken from the endosperm of corn. First isolated is the kynutin. And another very important, most of the students are getting confused. What is the naturally occurring abscisic acid that we all people studied? Abscisic acid is the zeaxanthin. Not to confuse, not to confuse. Abscisic acid is the zeaxanthin, which is the naturally occurring. Naturally occurring cytokinin is what, friends? Zeatin. Never ever get confused with that particular point. Okay. Listen, every one of you correctly. I have already told you that in the first class of the hormones only we discussed all the hormones having a particular derivatives. And when we are studying the cytokinin, I have told to all of you that cytokinin is actually derived from what, friends? Purines. If every one of you very quickly recall, I told you purines are adenine and guanine. Especially, friends, the cytokinin is actually derived from what, friends? The cytokinin is actually derived from the adenine and cytokinin is actually derived from the adenine. You know what is the other name? We can call the 6,4 hydroxy 3 methyl but2 enyl aminopurine. So friends, sometimes they can ask, they will give this particular IUPAC name and they will ask it is belonging to the which hormone. I am requesting all the students remember that cytokinin is actually a type of the aminopurine. Never ever confuse her with that particular point. This is the IUPAC name. I am requesting every student to remember compulsory. The cytokinin is exhibited in two forms. One is called as the transgiatin, friends, and another cytokinin, friends. So we generally call it as what giatin? Cisgiatin. What? One is called transgiatin, another one is called as the cisgiatin. I want you to compulsory remember. There is no point of remembering the structures you never get in the exam. Okay, now let's try to understand the most important where the cytokinins are getting biosynthesized. Once again, let's quickly recall. Auxin synthesized where? In the apical meristem. Is it? Yes, true. What about the abscisic acid? It is the ubiquitous plant hormone. What about the ethylene we discussed? Ethylene is actually a gaseous hormone. It is majorly present in the ripening fruits. If every student quickly recall, gibberellic acid is synthesized majorly in the seeds, and it is also plays a key role in the internodal elongation. So it is also synthesized in the internodes. Then friends, where the cytokinin is getting biosynthesized, I told to every student, cytokinin is going to show their action exclusively in the shoot. Cytokinin is going to synthesize their action exclusively in the shoot. Not to get confused with this particular point. So the question is very, very clear. If it is showing its action in the shoot, where the cytokinin is actually getting synthesized, friends, the cytokinin is actually getting synthesized, where, friends, cytokinin is getting synthesized exclusively in the root. Not to get confused with this point. Cytokinin synthesized at the root. From the root, how does it travel to the shoot, friends? That's also a very important one. Cytokinin, whichever synthesized in the root, it is going to travel to the shoot via xylem. Via xylem into the shoot along with the water and minerals. Not to get confused with that point. So I can tell to all the students that 90% of the cytokinin is getting biosynthesized in the root apical meristems only. Not to get confused with that particular point, friends. In addition to the roots, young maize embryos, young developing leaves, and young fruits also synthesize the cytokinin. I am requesting all of you, don't think that cytokinin is only synthesized in the root. It is going to synthesize in other parts also, but in these parts the concentration of cytokinin is extremely lower. Where the maximum amount of cytokinin is getting synthesized, friends, it is getting synthesized majorly in the roots only, not to get confused. Now once again, all the students has to remember the precursor. Why? Because friends, the moment we compare one plant hormone with another plant hormone, then only it is very easy to remember it for the long period of time. Now listen friends, let's talk about auxin. You remember what is the precursor for auxin? All of you remember, right? The precursor for auxin is tryptophan. What is the precursor for the gibberellic acid we studied? GGPP. What is the precursor for abscisic acid we all studied? IPP, isopentenyl pyrophosphate. What is the precursor for the hormone ethylene we studied, friends? We have studied the gaseous, the amino acid, which is the methionine. Same way, what is the precursor for the cytokinin? I want every student has to remember compulsory without confusion. It is called DMAPP. Easy to remember. Dimethyl allyl pyrophosphate. What friends? Dimethyl allyl pyrophosphate. Friends, dimethyl allyl pyrophosphate is how many carbon containing compound, friends? Dimethyl allyl pyrophosphate is the 5 carbon containing compound. Not to get confused with that point. Now I am going to explain to all the students how cytokine is getting biosynthesized. Every one of you listen to me, friends. You know what is the function of this particular DMAPP, friends? The major function of the DMAPP is transfer of what group? Every one of you listen. I'll ask you in the next class also. It is involved in the transfer of isopentenyl group. Now, friends, to whom it is donating the isopentenyl group? That is the important question. It is donating the isopentenyl group to the AMP molecule, not to get confused with that particular point. So all the students listen, this is called DMAPP. DMAPP, listen every one of you very correctly. It is donate this isopentenyl group to what, friends? To the AMP molecule. So you only look into this, friends. This is AMP. Where the isopentenyl group is added, here it is added. Upon adding isopentenyl group, AMP converted into what? AMP converted into IPMP. Very simple. To the AMP, we have added isopentenyl group, that's all. Then it become isopentenyl AMP. Now, friends, from the isopentenyl AMP, what we are doing, friends, we are removing this phosphate. You can see that. Upon removing the phosphate, it is converted into IPA, isopentenyl adenosine. Later, friends, from the isopentenyl adenosine, we are removing the sugar. Upon removal of the sugar, isopentenyl adenosine is getting converted into what, friends? It is getting converted into isopentenyl adenine molecule. Not to get confused. Now that IP or isopentenyl adenine converted into transgeatin. Then the transgeatin can be treated by a isomerase enzyme, and it is converting into what, friends? It is converting into cisgeatin. Not to get confused with that particular point. I want you to understand it very correctly. Once again, all the students try to listen to me very correctly without confusion. No need to confuse. It's very easy. DMAPP is this, you don't need to remember this. This is the other pathway. Remember one pathway enough. DMAPP, what it is doing, friends? It is donating or it is transferring the isopentenyl group. It is donating or transferring the isopentenyl group to whom? It is donating or transferring the isopentenyl group to the AMP molecule. Whenever it is donate isopentenyl group to the AMP molecule, what happens? AMP molecule become IPMP, isopentenyl monophosphate. From the isopentenyl monophosphate, which group is getting deleted, friends? There is a removal of the phosphate group. Upon removal of the phosphate group, isopentenyl monophosphate is converted into isopentenyl adenosine. Then isopentenyl adenosine, upon removal of the ribose sugar, it is converted into IP, which we can call isopentenyl adenine. Now that isopentenyl adenine, in the presence of hydroxylase enzyme, converted into transgeatin. Then the transgeatin can be treated by a isomerase enzyme. It is converted into cisgeatin. There is no point of remembering even a single structure also. Don't waste your time on remembering the structures. So what you have to focus on? You have to focus on the precursor. That is the DMAPP. Never ever get confused with that. Now, friends, the most important one where we are going to expect the questions mostly is, that is what, friends? That is the functions of the cytokinin. So friends, if you don't remember the functions of the cytokinin, you are unnecessarily losing the question. So it's very important to remember the functions. Listen, all of you, what are the major functions of the cytokinin? Anyway, I have already told you. Cytokinin function is what? It is involved in the shoot development because it is synthesized in the root. Let me tell you one very interesting point. So listen, it is synthesized in the root, it is traveling to the shoot, correct na? I have clearly explained you one important statement to all the students. Wherever the hormone is synthesized, it never show their action in that particular area. You remember, right, friends? Site of production, site of action is entirely different. Is it or not? So, friends, cytokinin synthesized in the root. So, where it will show the action? In the shoot. So, what I can say? Positive development in the shoot. No doubt about it. And in the root, what you can say that, friends? In the root, it is going to show the negative development. Not to get confused with this particular point. Positive development in the shoot, negative development in the root. Never ever get confused with that particular point. Okay, now listen to me correctly. So that is true. Positively in the shoot apical meristem, negatively in the root apical meristem. All the students understand this point. What is the interesting point? Suppose example, everyone listen. Suppose example, if the cytokinin genes got mutated. In other words, if the cytokinin genes are doing overexpression, then friends, you know what happen? Opposite will happen. What do you mean by the word opposite? It will start showing positive growth in the root, and it is inhibiting the shoot growth. When? Only during the overexpression. Not to get confused, friends, I am going to explain to all of you once again. Suppose, friends, hormone cytokinin, listen everyone, in the root, it will show negative growth, negative development. In the shoot, it will show the positive development. Every student understand that very correctly. Now, what is opposite? If there is a overexpression of the cytokinin occur, it will do opposite. What do you mean by that? Negative growth in the shoot and positive growth in the root. Not to get confused. That is only during the overexpression or in the during the time of mutations. Not to get confused with that particular point. That is the first point. Second point, every student listen to me correctly. The hormone cytokinin, listen all of you, the hormone cytokinin plays a very crucial role in the cell division. Now the question is very clear, how the hormone cytokinin is going to show the process of the cell division. That's a very important one. The hormone cytokinin is show the cell division by activating an important cyclin molecule. We can call it D type of cyclin. This also they can ask the question. What is the question they can ask? Which of the following cyclin molecules are getting activated by the hormone cytokinin? What do we call it as, friends? We call it as the D type of cyclin. All the students listen. All the students listen. If the cytokinin hormone want to involved in the cell division, there should be a activation of a different molecule. First, there should be an activation of CDC25 like phosphatase molecule. CDC25 will activate CDC2 kinase. CDC2 kinase will activate CYCD3. Then finally, friends, CYCD3 gene is going to express. What it is going to express? It is going to express, friends, D type of a cyclin. Not to get confused, easy to remember. Once again, I am explaining to all of you. First of all, cytokinin hormone will activate the CDC25 phosphatase. Then CDC25 phosphatase activate CDC2 kinase. Then CDC2 kinase activate CYCD3 gene. Then CYCD3 gene activate undoubtedly D type of cyclin. In simple words, the cytokinin is involved in the activation of D type of cyclin. In order to activate the D type of cyclin, first it is going to activate all this. Finally, with the help of D type of cyclin, cytokinin is involved in the cell division. Not to get confused and not to forget. And the most important significant function among all, now we are going to study. I have explained it to all of you many number of times, not to get confused with the question. In the exam, always hormones makes you confused, friends. In the exam, if they ask you a question, which of the following hormone promote the senescence? Undoubtedly, what is the answer you all people will give? You all people will give the hormone ethylene. Not to get confused with that particular point. Is it or not? Of course, yes. Yes, right. Now you listen my question. Hormone ethylene is promote the senescence, which delay the senescence. Undoubtedly, friends, the cytokinin is going to delay the senescence. It's really interesting. All the students has to understand me. Which means what? Cytokinin always help the plant to become green color for long period of time. Every student, please look into this. This is one plant. This is the second plant. The first plant, they kept under the cytokinin solution. The second plant, they kept under the normal solution. Whatever the plant, all the students follow me. Whatever the plant they kept under the cytokinin solution, it became green. I'm sorry. It became yellow after few days only. After few days only, it became completely yellow. But friends, whatever the leaf they kept in the cytokinin solution, it is green for very long period of time. First of all, every student has to understand very correctly without confusion. What it is, how the leaf turns into yellow. That's a very good question. Suppose, friends, this is a leaf, right? How the leaf can be converted into yellow, you all people has to tell me. You all know that, friends, why the leaf is green in color. First of all, every student know very well, because of the chlorophyll. Okay, because of the chlorophyll. Listen, because of certain RNA molecule, because of certain protein molecules, because of these three, leaf will be green in color. Now, you know what happened? There are certain degradative enzymes are there. I want every student has to follow me, friends. There are certain degradative enzymes are there. They will destroy the chlorophyll. They will destroy the RNA. They will destroy the protein. So now all these are all lost. Then the leaf is going to convert it into yellow. You got the concept correctly. Now that is not the point. What the cytokinin is doing, that is the point, right? All the students listen. Cytokinin will come and block this enzyme. Which enzyme? Degradative enzyme. Remember my words. Whenever the hormone cytokinin block the degradative enzyme, immediately what is going to happen? When the cytokinin block the degradative enzyme, then the chlorophyll will be active, RNA will be active, protein will be active. Friends, then the leaf never ever turns into yellow very immediately. Once it become matured or older, then only it will become yellow. That's why. So it is delaying the senescence. That's why, friends, the hormone cytokinin is called as what hormone? Anti-aging hormone. Cytokinin is called aging hormone, whereas ethylene is called as what hormone, friends? Ethylene is called as aging hormone. Never ever get confused with this particular point. Anti-aging and followed by the aging. Don't confuse, friends. Then the next one, the hormone cytokinin inhibit apical dominance. So what's the point? What's the point here? The hormone ethylene and cytokinin are antagonistic to each other. Such an important statement it is. Antagonistic to each other. With respect to which function? Leaf senescence only, only in this function. This experiment was done by a scientist called as Richmond. That's why this effect is also called as a Richmond-Langerhans effect, not to get confused with that point. So, you all people remember, right? Which hormone promote the apical dominance? Auxin. What we discussed, friends? If auxin is present at the shoot, what we discussed, it never ever allow, what it never ever allow, it never ever allow the growth of the lateral buds or the lateral branches. This is what we studied, right? Friends, listen to me very correctly. Whenever there is a presence of the cytokinin, you know what happened? Cytokinin is suppressing the level of the auxin. As a result, friends, there will be a growth of lateral buds. So what I can tell you right now, I can tell you that hormone auxin, hormone cytokinin is antagonistic to each other, because like this only you will get the question which makes you confused. Antagonistic to each other with respect to which function? Only the apical dominance. Whereas a hormone ethylene and cytokinin are opposite to each other only with respect to what? Leaf senescence. Very, very important function it is, not to get confused with that point. Another very, very, very important where you can get a twisted questions, that is called morphogenesis. All the students has to listen to me correctly. I already told you, wherever the hormone cytokinin is present, friends, the hormone cytokinin is always going to stimulate which initiation? Shoot development or the bud formation. So what I'm trying to explain to all the students, suppose you take a culture. If you take a culture, if you keep low concentration of auxin and high concentration of cytokinin, what it is lead to the development of? Undoubtedly shoot. Yes or no? Suppose if you keep high concentration of auxin and low concentration of cytokinin, what does it leads to the development of? It leads to the development of the root, because auxin is present more. But friends, what most of the students will get confused is, if you keep both the auxin and cytokinin in 1:1 ratio, what does it will develop? Most of the students will give the answer that it will develop both root also, it will develop both shoot also. So what you have to remember, this is wrong. If you keep both in 1:1 concentration, don't think that it will give both root and shoot. It will give to the callus. What is callus? Undifferentiated cells, undifferentiated mass of cells, a group of cells, a bulk of cells which are undifferentiated, that is called callus. Why? Because we kept both in equal concentration. Suppose if you increase the auxin, once again root. If you increase the cytokinin, once again shoot. Not to get confused with that particular point. I want you to understand that correctly. More auxin root, more cytokinin shoot. If both are equal, it is going to give rise to the callus. Not to get confused with that, friends. Then the another very important function performed by the cytokinin, most of the students will skip this function also. Very good function. It is very important function. The hormone cytokinin involved in the expansion of leaves also and cotyledon also. My friends, so once again, they have done the experiment in the Arabidopsis plant. They have taken the leaf of the Arabidopsis. They have taken the cotyledon of the Arabidopsis. T0 represents the 0 days, starting time. 3 represents 3 days. You only look into this, friends. Till 3 days, you have not treated the leaf with cytokinin. So here cytokinin is not there. You tell me, is there any expansion of the leaf? Never, ever. But friends, when you take the leaf and if you treat it with the cytokinin, you see this cytokinin is involved in the leaf expansion. Friends, please don't get confused with that. Same with the cotyledon. So if you keep only cotyledon without cytokinin, there is no expansion because no cytokinin. But after 3 days, when you keep cotyledon with giatin, that is the cytokinin, there is an expansion. So what is the conclusion? Cytokinin plays a key role in the expansion of the cotyledon, in the expansion of the leaf also. How? By the process called enlargement of the cell. Friends, what are the major and very significant function all the students has to remember? Cytokinin hormone plays a crucial role in converting etioplast to the chloroplast. This is etioplast, friends. This is what? Chloroplast. Question is very clear. How does the etioplast is getting converted into the chloroplast? Just listen everyone. See, friends, etioplast, you remember, dark grown. Chloroplast is what? Light. Compulsory light is required. So if the etioplast want to be converted into chloroplast, what has to be synthesized? That's the significant question. There should be a synthesis of the chlorophyll molecule compulsory. That's an important point. So they can ask a direct question in the exam. Which of the following plant hormone is involved in the synthesis of the chlorophyll in the dark grown seedlings? Which is the cytokinin. Never confuse, friends. Cytokinin promote the conversion of etioplast to the chloroplast by stimulating the synthesis of chlorophyll. So friends, what are the functions? Let's quickly understand. Cytokinin is involved in the positive growth in the shoot, negative growth in the root. If the overexpression or mutation occur, it will show opposite. Cytokinin involved in the cell division by activating D type of cyclin. Cytokinin delay the senescence. Cytokinin inhibit the apical dominance. Cytokinin majorly involved in the shoot development. Cytokinin is involved in the synthesis of chlorophyll. Cytokinin is involved in the leaf and cotyledon expansion. Not to get confused, friends. Many times the question from the cytokinin they will ask by combining with other plant hormone. That's why you have to remember the hormones very clearly by interacting with the other hormones. Never forget it. Okay, that's all for now. I'll see you in the next class. Okay, thank you all.